Hi there. Thank you for taking the time to check out the research I'm undertaking at the University of Sydney as part of my honours year. Feel free to pause the video at any time if you would like to spend more time on a specific section. My collaborators in this project identified a novel pain-regulating gene called SETDB2. They recognized mutations to this gene in humans completely abolished pain perception, and SETDB2 knockout mice had significant pain and sensitivity. However, the effect this mutation has on the structures of the pain pathways is still not understood. My project stems from this research, where I aim to identify structural changes and neuronal morphology in the thalamic region responsible for relaying pain from the body to the cortex in pain-insensitive SETDB2 knockout mice. I hypothesize that knockout of this gene will result in abnormal neural cytoarchitecture and cell morphology, specifically within the ventroposterior thalamus, as it receives all incoming painful sensory information, and disruption to these incoming signals may consequently result in pain insensitivity. SETDB2 plays a regulatory role in two key mechanisms involved in early development of the brain and the pain pathways. The first mechanism is the role of SETDB2 to regulate the size and location of anterior cortical areas by suppressing fibroblast growth factor 8, or FGF8. FGF8 is defined as a morphogen, and its expression develops brain structures by signaling radial glia to extend. SETDB2 suppresses the signal when the brain is fully formed to prevent overdevelopment of the brain. SETDB2 is also involved in gene repression by trimethylating the protein H3K9, known as H3K9ME3. Chromatin marked by H3K9ME3 modifies its structure to be densely packed heterochromatin, which prevents transcription factors from accessing the DNA. The formation of this transcriptionally silent heterochromatin is essential for gene regulation, the maintenance of chromosomal integrity, as well as normal cellular identity. Now, to identify potential structural changes, I visualized the structures of interest using multiple stains. The first stain I used is parvalbumin, which stains fast spiking interneurons, which are the most abundant type of inhibitory interneurons in the brain. Additionally, the positive staining of parvalbumin allows for the identification of the ventroposterior thalamus, or VP, and the reticular thalamus, or RT. Another stain I used is neuin, which specifically stains for neurons, and thinin, which stains for negatively charged nucleic acid. Now, in terms of data acquisition, parvalbumin allowed me to calculate the area of the VP thalamus by tracing its border, indicated by the dashed ovals. Neuin and thinin allowed me to count individual neuronal cells at high magnification, identifying any abnormal cells within the VP thalamus. My first main finding is that out of all the cells counted, 25% of them were abnormal in the wild-type mice, whereas 70% of them were abnormal in SETDB2 knockout mice. The graph on the far right shows the average number of morphologically normal neurons in blue and abnormal neurons in red, from the front to the back of the VP thalamus in both wild-type and SETDB2 knockout mice. Secondly, the neurons of SETDB2 knockout mice have enlarged somas. Two neurons from the wild-type and the knockout group were circled, showing the disparity in soma area. Finally, there was no significant difference in the VP thalamic area or in the total number of neurons counted between these groups. So to tie everything together, SETDB2 is responsible for normal brain formation and regulating the cell cycle. Knockout of SETDB2 in mice leads to significant pain insensitivity, but has no effect on the area of the VP thalamus or the total neuron count. However, SETDB2 knockout mice do have significantly higher number of neurons with an abnormal morphology compared to the wild-type mice. These results suggest that normal neuronal morphology in the VP thalamus is critical for normal transmission of pain from the body to the cortex. Thank you for listening and for your interest in my project.